So hi everyone. It's good to see so many people joining. So we will just wait a few more minutes to let everybody in here.
It's a little bit like a personal handshake for everybody.
Yeah,
so,
yeah, but we see many
everyone who's joining,
names.
welcome. So many people are attending. That's a good sign.
Yeah, so we're really happy about this. Yeah, and we see actually many familiar names, but some new ones too. So we're really happy about that, and it's good to connect here. So yeah, so I think we can start really slowly and people can still join. So yeah. Well, yeah, shall we start, Dirk? Yeah, I think it's good.
Yes, go ahead, Rebecca, please.
Yeah, so yeah. Hi everyone. So we are really glad to see as many people joining, and good morning and good afternoon. So wherever you are, we welcome you to our webinar, GMP Radiolabeling Development from First Discussion to Method Transfer. So my name is Rebecca. Just a short introduction. I'm working here as a senior sales representative at COP for 2.5 years now, but joined the radiopharma world almost 20 years ago. But that's also a first for me today. So I'm also excited but really happy about this event. And I will be your host for today's session. And we are very pleased to have a strong program lined up for you, combining external perspectives, strategic insights, and also give you some insights on our practical experience from our scientific team. And I would love to start out with a short introduction of our speakers, and then I will also give you some details on today's agenda. So to begin, I'm very pleased to introduce our external speaker, Erland. We are really happy to have you, and he is an expert in the radiopharmaceutical development and GMP regulated environments. He has extensive experience supporting complex programs from early stage development to clinical manufacturing, including method validation, technology transfer, and CDMO management. And next, I'd like to introduce Dirk Freitag-Stechel. He is our CEO. He is also co-founder of TRIMT, and he has deep expertise in pharmaceutical analytics, GMP-regulated environments, and radiopharmaceutical development. And he has been actively involved in the development of novel radiotracers and radiolabeling technologies, and is bridging early research and routine quality control. His core strength lies in building high-performance laboratory structures and supporting complex development projects in the field of radiopharma. And I'm also happy to have Falco with us here today. He leads the analytical chemistry department at COP Contract Labs, and he has a strong focus on radiolabeling, radiopharmacy, and nuclear medicine. He is very experience in method development, validation, and transfer in GMP environments, and particularly for complex and high-risk pharmaceutical projects. And rounding off our speaker panel is Konstantina Makripidi. She specializes in radiopharmaceutical research with expertise in radiolabeling as well as analytical method development and optimization. She works closely in the transition of early stage research into robust validated methods suitable for GMP environments and routine application. So that were a few words to our team here presenting today, and and now to our agenda. So we will have about 45 minutes of presentations, and this will be followed by a 50-minute Q&A session. So I would already like to encourage you now to submit questions throughout the webinar via the Q&A or F&A section. So we will have a look at those questions later on, and we will also share, just to let you know already now, this recording, and we will also share the questions alongside that after our webinar, so you won't miss out on anything, even if we not make it to address these questions due to the time. And we'll be focusing on radiolabeling from multiple angles, including its relevance in early drug development, method development, and optimization. And our goal is not only to provide you today with scientific insights, but also to give you a better understanding of how these approaches can be successfully implemented and transferred into real-world projects. And we will begin with a brief introduction of COP, followed by insights from the industry perspective, as well as detailed discussions on an integrated analytical approach and practical insights from development through to GMP execution. So now that you know what we do today, we would actually like to begin. And so to kick things off, I'm really happy to introduce Dirk, who will give us a short presentation of COP Contract Labs.
Yeah, thank you, Rebecca. Hello, everybody. So I keep it really short. And I will encourage you. We are at the SNMMI in Los Angeles this year, and also at the A&M with a booth. So if you need some further information about COP Contract Labs, what we are doing, what we are planning, just reach out to Rebecca immediately if you want, or contact us and let's meet in US or in Vienna. I would never have imagined 20 years ago, this is when I started my career of COP Contract Labs, to give a webinar with English with so many skilled people in the auditorium from all over the world. So thank you for that. And that's due to our development as COP Contract Labs from a small environmental lab at the end of the world in Eastern Germany to a really special service provider for radiopharmaceuticals. So we stand for a better future, and we stand for support. We support startups, but also manufacturing, also big companies, and we want to bring them next generation pharmaceuticals fast and reliable to the patient. We deal with narcotics, orphan drugs, high potent drugs, but especially with radiopharmaceuticals. And we have a long experience in that, as we are here part of the Radiopharmaceutical Valley, like we call it. The new name is Nuclide. We are located in Saxony near Dresden, and global players, let's say global players like ABX and like Rotop are next to us. We have a strong leadership experience and beautiful facilities. You can see here the people from COP. Therese is leading our operations. You will meet today Falco telling you something about our newest service. And we have also a very strong microbiological lab led by Marco. In total, we are 70 people working with COP, scientists, technicians, all very skilled people. Next slide, please, Rebecca. Yeah, we invested a lot over the last 2 decades in really modern labs. And if you want to visit the Radiopharmaceutical Valley or the Nuclide area, please step by. We love to show it to you. We have a really state-of-the-art building. What we have patented is the sterility test of radioactive pharmaceuticals with membrane filtration. And we have all analytical techniques you need to bring a radiopharmaceutical fast and reliable to the customers in place. And what's important to mention, we are a contract lab. We invested in a contract lab. We are completely independent, and we will never be a CDMO, but we want to speed up the process with CDMOs to bring the medicine fast to the client and to the patient. Next slide, please. This is what we do in our analytical division, as I mentioned, led by Falco. Today you learn about our radiolabeling services. We are very good, and to the best of my knowledge, the only lab worldwide who's doing hot, that means radioactive studies on leachables, extractables. We are able to identify visible particles also in radioactive formulations, and we are quite good in impurity identification, method development, method validation, stability studies, nitrosamines, also hot, and we do also hot stability studies. Next slide. What makes us very special? I mentioned it. There's only one lab worldwide where you can get a sterility test with membrane filtration under GMP, and that's us. What we are also doing is bioburden testing. We look for bacteria endotoxins and we identify microorganisms. And it's very difficult to ship radiopharmaceuticals. That's why we invented a cassette-based sterility test and licensed it to Traces. So if you look for Sterinow, it's another webinar, but if you look for Sterinow, you will see the solution at Traces. And please feel free to reach out if you need some more information on that. What we are currently working on is the world's first radioactive monocyte activation test. As most of you may know, is that the test for pyrogens, the so-called rabbit test, is forbidden in the EU. And we will have by summer this year the world's first monocyte activation test in place. And this brings you to what we are doing. We take techniques from the classical pharma, where we have long experience, and bring it to the field of the radiopharmaceuticals. And with that, our goal is to assist really skilled people, great companies, to bring medicine faster to the patients. And it's my great pleasure to introduce Erland from Actithera. Erland is one of these skilled people, and I hand over to Erland. Erland, stage is yours.
Thank you so much, Dirk. I'm really honored for this opportunity to provide the industry perspective and exchange on GMP radiolabeling development. And yeah, to everyone participating, thank you, and good morning, afternoon, and evening. In my presentation today, I will focus on radiolabeling, where it becomes challenging in the real world development, because these, the science behind mechanism of mode of action for these products, and also even the radiolabeling process is relatively simple to target, to label your molecule, target molecule with the isotope, sterilize it, dispense it, and deliver it. But yeah, my experience, and I'm sure yours also, is that these are really highly complex pharmaceutical products to develop, and will have a lot of different dependencies. That creates a lot of challenges to overcome. And you mentioned it already, speed is an essence, and CMC is always under pressure. And so it's also, I appreciate the opportunity today to really connect and collaborate and share learnings across the community, because this increases our overall chances of success. Because we all have a joint mission to bring really novel radioligand therapies to patients, cancer patients in need. But yeah, just quickly on my background, since I'm filling the screen here. So I recently joined Actithera as CMC development director, and I have about 10 years in developing radiopharmaceuticals in analytical process development and also manufacturing. And I worked, as you see, across different modalities and isotopes, and also really important across different environments. So internal labs, CROs, and also CDMOs. And this is important because I think it's especially in these kind of cross-functional settings and intersections that many of the challenges we have becomes visible. But yeah, before diving into these challenges, I will give you a quick intro to Actithera's innovative science. So if we go to the next slide, please. So Actithera is a discovery company that is designing and developing next generation radioligand therapies for difficult to treat cancers. And we focus on modified peptides and small molecules that really increase tumor retention time and by that minimize also radiation exposure to healthy tissue. And our core technology that we apply is covalent targeting, where we really target, the targeted molecule is irreversibly cross-linked to the target, which prolong the radiation dose. And on the next slide, you can really see a real-world example. So we have a few SPECT images where we are comparing our lutetium-177 labeled Act319 FAP compound on the right side with Novartis' most advanced FAP compound on the left side. So you can really see this favorable tox profile and also high prolonged tumor radiation dose over time, which really gives this therapeutic window that is so important. So, but again, we are looking here at the preclinical data, and you all know that this doesn't guarantee a smooth CMC development to a clinical study, because actually our experience is that the more complex the molecule you have, the more sensitive the radiochemistry often becomes. So on the next slide, let's dive into this and start to talk about some of the challenges and where they begin. So, I mean, immediately after you are radiolabeling your precursor with an isotope, the radioactivity leads to radiolysis. So you're forming this oxidized species that can really degrade and has an impact on your product impurity profile. And often you see that this radiolysis effect is not linear, so it's not so even at higher radioactive concentrations you see different correlations than what you do at lower activities. And also this decreasing radiochemical purity over time, it really becomes a logistical issue for these products that already often have a short shelf life and thereby also so important for the usability of these products to really be able to distribute it and deliver it to the hospitals and the patients. And also radioligands, you very often see a high precursor and also formulation sensitivity. So really small changes can have a dramatic impact on your labeling yield and purity. And also you see this tendency that while scaling up, you had success in your lab setting, but when you are going towards clinical batch scale, then suddenly you have unexpected results of batch failures. And another major challenge is the lack of robustness across environments. So really again talking about comparing an internal lab where you maybe have good results, and then it comes to a CRO or a CDMO, and there are different outcomes. And also I've experienced such outcomes could be driven by simply that the equipment and method variability, so that there are important differences that you maybe don't see immediately that will give inconsistent result and these kind of late surprises. So one example on the right hand side here with the chromatogram where you discover that your precursor is really not 98% as the original analysis was telling you, but you discover later that this was less than 90% with higher resolution. This is, of course, the later you make these optimizations, the worse or the bigger the impact is. So yeah, the take-home message here is really that radiolabeling is not a fixed process. It's highly context dependent and sensitive to real world variables. And differences in equipment, workflow, operator handling, it really matters to create robustness. And if your process and methods it's only working in one lab, I would say it's not ready actually. So so on the next slide, let's go a little bit more into this kind of upscaling. So we're going from lab success and how do you transfer this to, or what can be challenges transferring this to GMP validation, tech transfer and manufacturing. Because here, when you take this step, here is often when you get the surprises or see the gap. So instances that could lead to this is typically that your process validation has a too narrow, or your process has a too narrow window. So when maybe you make smaller adjustments in a higher scale, then you really it's difficult to demonstrate this robustness that we need in a clinical setting. And then often you could see suddenly batch to batch variability in this setting when you are testing a clinical scale. And also, technology transfer. So really these the more complex methods and also not robust methods, radiolabeling process, then there is I would say a higher risk, of course, to have a failure in a CDMO setting. And sometimes you have this kind of hidden dependencies or critical parameters that you didn't test or you didn't investigate that is really occurring when you are scaling up into GMP setting. I would also say that typically to apply process and methods that are still under development and not locked, that can create challenges when working, especially in this parallel setting across sites, and of course we talked about already the variations with the equipments that can really lead to inconsistencies. So the GMP setting is really a different ballpark or different level, and so again, having success in a lab is not proof that you have a GMP that you are GMP ready. And my experience at least, that the failures are very not often scientific, but it's more based on the operations and lack of process understanding or control failures. So next slide, please. So let's talk about what really is required for CMC readiness. So, and what does it actually mean? So what is important is to have a deep understanding of your radiolysis, of your process, your methods, and your radiolysis mechanism, the impurity profile, and that these are really properly characterized systematically. That's said by a very early defining or disclosing critical process parameters and determine on your quality attribute targets to measure these. So really to have these standardized methods as early as possible, especially HPLC, and try early to create the reproducibility across sites and different equipment, and also operators, to see how robust is it really. So the ideal scenario is, of course, to have that you have designed and developed and locked a robust analytical method and process early that is well understood and scalable and really delivers this safe and the same product for every batch. So we're really looking for this predictability and reproducibility. And on the right-hand side here, you see a very nice tool, which is from Design Experiment, that can really be used, especially for this, for instance, for formulation strategy testing, and really understand your process. But so the next slide is really just summarizing these kind of points, and maybe in a more practical manner. So So to summarize this, it's really some recommendations from us is to lock your analytical methods early as possible, and then of course test clinical relevant conditions, so not only small scale but large scale from the very beginning. Design really and have this mindset that you are really to transfer the method and process to another place and a GMP setting, and then I can't overstate really investing heavily in formulation development. So this can be provide a lot of also it's a good investment for later when there you also maybe have a pipeline and can sometimes apply your formulation to different products, or it survives process and method changes. And of course, as we talk about today, to really use expertise across and do parallel testing to show this robustness early. And I would say also always apply this systematic experimental design and data collection, like applying a DOE in an early phase also helps to maximize the value of the data you are creating. So to summarize everything, just I would say that successful RT development programs, they treat radiolabeling more as a system, and that is really reliant on real-world variables and not single steps. And the big risk is that you have early success that is not really reproducible in a real-world setting. So it's really to bridge this gap. It requires robustness and scalability and focus on transferability really early in your development. So this was what I was planning to say today. So thank you so much for listening, and then I'm happy to take questions later. Please.
Yeah, great. Thank you, Alan. That was really a great presentation here. We're really happy that you shared your insights with us. And and with that said, I would like to hand over to Falco, our head of the Analytical Chemistry Department, who will lead us through some key aspects of radiolabeling and general considerations.
Yes, so hello everyone. Again, from my side, thank you, Rebecca, and thank you, Alan, for presenting your perspective on this radiolabeling aspect. And I think what we have learned is that radiolabeling really is a critical development step. And now we want to present our perspective on key challenges we typically face, because radiolabeling directly impacts validation, transfer, and quality control from the beginning to GMP testing. And from our experience, typical customer projects face such topics like short half-lives, tight timelines. They're complex, and we have sensitive labeling chemistries. At the beginning, sometimes there's unclear analytical requirements, and we have an increasing GMP and regulatory expectations. So that is why radiolabeling really requires an integrated approach from the beginning. And with having that said, we define the right path from the start. What means we ask the key questions to our customers in a very, very early stage. So we face the problem. We will ask, is the chemistry robust? Does it require any optimization? Which analytical methods are required? Is anything known? How should or what should the data package at the end look like? What are the concrete expectations for transfer and project outcome? What is the timeline? Which milestones are realistic? What timeline is realistic? And how can GMP requirements be met from the beginning? And we have learned and we know that without having those answers early on, projects really become inefficient and difficult to transfer. So how do we solve this problem? We understand ourselves not as a step-by-step, but as a whole life cycle service provider, as a strong partner at your side, and offering an integrated approach to development and GMP execution. We will start every project at the stage of a scientific consulting. We will then perform the radiolabeling development, afterwards the analytical method development, then the method validation, and finally routine GMP testing and also batch release. How do we do it? Because high impact science requires exceptional people, which is obvious. And we have some, don't want to say hurdles or obstacles, but there's several things we have to navigate. We have demanding radiochemical processes, which I already said. But from a regulatory side, we also have strict radiation protection requirements. We have regulatory complexity and technical hurdles also connecting to GMP requirements. And of course, working with radioactive materials is always a significant investment in time, infrastructure, and also expertise. And how do we succeed on this way? We have strong collaborations across the disciplines. We have a very deep scientific expertise. We share our responsibilities and commitment in a very efficient way. And we have a team culture which is built on trust and excellence. And at this point, I am more than happy to hand over to one of these excellent colleagues in our company. And I want to introduce Konstantina, who is one of our radiolabeling and method development expert and responsible for many of our projects.
Thank you, Falco, for the introduction. And hello, everyone. I'm very excited as it's my turn now to welcome you to our webinar. Today, I will guide us through a workflow, a step-by-step workflow, starting from the first radiolabeling tests to routine GMP quality control testing and product release. This workflow can be separated into 3 phases. In the first phase, we start with the scientific consulting, where key decisions are made and the overall project scope is defined. We also plan and test the first radiolabeling approaches, and we proceed with the analytical method development. Before moving to phase number 3, all methods, the radiolabeling and the analytical ones, need to be optimized to achieve the highest possible efficiency. In the third phase, the methods are validated to align with the GMP quality control standards, and they are transferred later to routine quality control testing in GMP environments and product release. Next slide, please. Thank you. In this very first stage of development, the scientific consultation takes place where we define realistic timelines and the development scope. We provide guidance on the necessary analytical strategies. We evaluate the precursor structure and its chemical properties, as this is especially important for the labeling approach that we will select, because we want to have a high efficiency. And when we evaluate the precursor, taking into consideration the whole system, meaning the molecular target, the linker, and the chelator, we can ideally avoid or minimize any degradation products or any decomposition of the precursor during the labeling, that it will result later in a reduced purity. Depending on the project scope, the chelator, and of course the intended application. Next slide, please. The most appropriate radionuclide is selected among the allowed isotopes we can handle here in CUP. Of course, all radiolabeling procedures need to align with our capabilities, with our handling capabilities. I'm sorry, in the previous slide. Thank you. Yes, which the handling capabilities are dependent on the isotope chosen, and they are varied, for example, from 50 gigabecquerels per year for actinium-225 up to 50,000 gigabecquerels per year for technetium-99m. Now we go to the next slide. Once the very first key questions have been answered, we have a deeper look into the labeling approach and to the analytical needs, as we should select the most appropriate chemistry for our ligand isotope combination. We also define the analytical strategies that are needed, as this is very important to do it from the beginning. It's very important to have the right analytical concept from the beginning of the development, as we want to monitor the labeling procedure and to evaluate the quality of the product from the early stages. When the chosen labeling method is then tested and is optimized to achieve the highest radiochemical efficiency, radiochemical yield, and radiochemical purity, resulting in a robust and reproducible radiolabeling process. The radiolabeling procedure can be optimized by varying different conditions like the temperature, the reaction time, the pH, the buffer composition, the precursors concentration into the labeling solution, the order of addition of the different components of the reaction, as well as by adjusting the purification process after labeling when it is applied. Next slide, please. But now let's see a real example from a method, from a labeling method optimization. In the first chromatogram, we see that when the reaction takes place at 25 degrees Celsius for 30 minutes, we obtain our labeled compound with only 6% of radiochemical conversion. By increasing the temperature to 70 degrees Celsius for 30 minutes, the radiochemical conversion is increased as well, reaching 62%. And next slide. Further increase of the temperature and the reaction time to 100 degrees Celsius for an hour resulted in the highest radiochemical yield and purity for our complex, which it was 96%. In parallel to the radiolabeling method development, the analytical methods are developed and optimized to enable us to monitor the efficiency, the impurities, and the purity of the product, the identity of the product, as well as its stability. The main analytics that are used are radio-TLC, radio-HPLC, LCMS, and in a later stage, ICP-MS and GCMS to quantify the metallic impurities or assess residual solvents respectively. Next slide. Thank you. During the method optimization, we come across many challenges that we need to overcome to ensure reliable and reproducible results. Some of the most common challenges we face are the coelution of impurities, the instability of the compounds, and problems with the matrix of the sample. Through the adjustment of the gradient and by changing the chromatographic conditions or by testing different column types, we can solve coelution problems or improve the peak resolution. Via optimization of the sample preparation and the dilution process, we can improve the stability of the sample or reduce any matrix effects. This way, we ensure that the developed analytical methods are robust and fully reproducible. But how these analytical methods optimization are translated into real data? So now we have here two radio-TLC chromatograms of the same sample. The difference is that the TLC plate was developed using different mobile phases. The second TLC chromatogram comes from a TLC plate that was developed using a mobile phase that had a higher percentage of the organic part. This change resulted clearly in a better separation between the peak of the labeled compound and the peak attributed to the uncomplex species. This example shows that even when a radiolabeling procedure has a pretty satisfying radiochemical conversion, can look like a poor result if the proper separation is not achieved, meaning if the proper chromatographic conditions, aka a proper analysis, is not applied. Let's move on to our next example, which is from the HPLC analysis. In the first chromatogram, when the initial chromatographic conditions are used, the result is a poor peak resolution for the two stereoisomers of the same labeled compound. After playing around with the temperature of the column oven, we managed to improve the peak resolution, and the result was that we obtained two distinctive peaks for each stereoisomer, meaning that we improved the chromatographic profile of this method. Next slide, please. Once the analytical methods are optimized, they need to be validated according to the ICH-Q2 guidelines and in alignment with pharmacopoeias to ensure that they are suitable for routine use in GMP environments and reliable release. The ICH-Q2 guidelines define, I would like to stay to the previous slide. Thank you. The ICH-Q2 guidelines define individual parameters for the validation of the methods, but for clarity purposes and for this presentation, we categorize the validation parameters into three key areas, which are the identity and selectivity, quantification, and reliability. So now let's go shortly through them. For identity and selectivity, specificity is assessed. Specificity ensures that the target compound can be surely detected and can be clearly distinguished from the impurities. It also proves that there is no interference from the matrix or the excipients that are present. We evaluate specificity by analyzing blank samples, placebo, spike samples, and standards. Next slide. To further demonstrate the specificity, we perform the force degradation study where we evaluate how the compound behaves under stress conditions. We practically generate the degradation products, and we prove that the method is suitable enough to reliably detect the degradation products, identify them, and separate them. We performed the degradation study by exposing the samples to stress factors like the heat, the light, oxidation agents, and alkaline or acidic pH environment. For quantification, which is the second validation key area, we assess linearity. Linearity ensures that the analytical response is proportional to the concentration. It is defined by creating calibration curves in different concentrations across the whole working range. Moving on to the second validation parameter of this category, the range, defines the working interval in which the analytical method is reliable. It proves that the method performs both at low concentration levels but also at higher concentration levels. The range is derived from linearity, accuracy, precision, and LOQ measurements. Let's go to the next slide. Thank you. Speaking of which, yes, the LOQ, which stands for limit of quantitation, defines the lowest concentration that can be measured with acceptable accuracy and precision. This is especially important when we want to quantify impurities at low concentration levels. The LOQ is calculated based on signal-to-noise ratio or using data derived from the calibration curves. Another validation parameter, yes, it's the precision. Precision ensures that the method is reproducible. It simply proves that that repeated measurements give consistent results. We evaluate precision by performing repeatability and intermediate precision studies. During the repeatability study, the repeated measurements are performed under the same conditions, while during the intermediate precision study, the repeated measurements are performed across different days using different devices and performed by different operators. When these studies are finalized, we evaluate and compare the relative standard deviations of the generated data. Another validation parameter of the third validation key area, which is reliability, is accuracy. Accuracy confirms the correctness of analytical results. It simply reflects how close are the measured values to the real ones, to the real values. We evaluate the accuracy using recovery studies with spiked samples across the working concentration range. And moving on to robustness. The importance of robustness was very nicely mentioned and explained by Erland earlier, but allow me to underline it again, as it is it's very important during the scale-up, during the method transfer into routine use, and during when the methods are tested across different sites. Robustness assesses whether the method is sensitive and stable in small changes of the method. We typically test robustness by varying some method parameters like the pH, the temperature, the composition of the mobile phase, or by applying some small changes in the stationary phase. Then we evaluate the impact that these changes have on the analytical results. In this way, we define the limits of the method, and we ensure that the method is sensitive and stable enough within these changes. And this ensures also that the method can have a reliable performance into routine use. Last but not least is stability. Stability is the last parameter, validation parameter we're going to refer to today. And it assesses how stable the labeled compound is over time. And this is very important for a reliable method. We evaluate stability by storing the labeled compound in different conditions and evaluating its radiochemical purity across different time points. We can also store the precursor in different conditions and then perform labelings and evaluate the radiochemical purity across different time intervals as well. So all validation parameters come together to ensure a reliable identification, quantification, and control. This enables a robust and reproducible analytical performance. When the methods are validated, meaning that they are meeting the acceptance criteria set for its validation parameter, they are ready to be transferred to the next step, which is their use in routine quality control testing in GMP environments and release. The quality control testing in GMP environments usually includes the identification of the labeled compound, the evaluation of its radiochemical and chemical purity, the appearance test, and pH measurements. We also perform trace elemental analysis, and we assess the residual solvents. The results of the quality control testing should be within the specifications defined during the validation of each method. Based on these results, we ensure that the batch meets the quality standards, and every batch that complies with them can be released. In this way, we guarantee that the batches reaching the patient are a safe and reliable product. So bringing everything together, a successful radiopharmaceutical development requires not only robust radiolabeling chemistry, but also strong data-driven analytical development and GMP-ready and validated methods. On the one side, yes, we need the robust chemistry and the analytical foundation, but on the other side, we also need an integrated and structured approach from the beginning of the development to validation, and finally to routine quality control testing in GMP environments. And when all of these are coming together, the result then is one and simple: a reliable and compliant batch release every time.
So thank you, Konstantina. This was a really great presentation, and I would like to take the opportunity to thank all of you. And first of all, thank all of you for joining and for staying with us during this interesting webinar. And also a big thank you to all of our speakers here. And when I look at the time, I think it's good to now move on to our Q&A session. So we received some questions, and just a little reminder, you can still submit questions. Please use the F&A or Q&A area if possible, and we will, if we don't address questions right now, we will definitely include them to the summary alongside the recording of this webinar we will share afterwards. So, and I think we have received a couple of questions via the chat, which will Dirk address. So,
yes,
yeah.
Am
Do
I
you see
allowed
them?
to
Yeah.
speak? Yeah, I see them.
Yeah, of course.
There was one important question. So the question was to elaborate the differences between a contract lab and a CDMO. To make it simple, a CDMO is producing clinical doses for a sponsor or for a partner company. And we help CDMOs to speed up this process. So we will never produce any clinical dose or any dose in our labs, but we help them to shorten the time for method development, method transfer, method validation, to bring medicines faster to the patients. So we are natural born partners of CDMOs, something like an extended quality control for CDMOs. And the other question is a little bit connected to that. Do we perform aseptic process validation runs as part of process validation? As we do not produce something, we cannot do process validation in-house, of course. But with our services like sterility testing, for instance, or maybe also like leachables, extractable studies, we of course can assist partners, CDMOs, manufacturers in that aseptic process validation runs. And again, I want to highlight the Sterinow cassette of tracers. Maybe this is a tool which can be used also for that.
Thank you, Dirk. So we have actually received a couple of more questions. So one of them was, and I think, Konstantina, you might be the one answering that. What happens if I want to use a new nuclide that is not yet covered by COP's regulatory approval?
Yes, that's a very relevant question, and we have received similar requests in the past. I can safely say that the process is very smooth. In the past, the similar requests we have received is either to add a new nuclide in our license or increase the activities of a specific nuclide as the project needed labelings with higher activities. But we handled that very efficiently without any complications, as we maintain a very strong and well-established relationship with the local authorities, which it allows us to adjust our allowance, our permitted allowance, easily without problems.
And
in a very timely manner. I'm sorry, Mr. Dirk. Yeah. Yes, if I remember correct, the process was done within a few weeks. Yes, sorry.
Yeah, Konstantina was perfectly correct, and I just wanted to highlight that this is something which makes the Radiopharmaceutical Valley nuclide in Saxony very special. We have extremely skilled regulatory authorities from the GMP side, but also from the radiation protection side, as there are so many companies and research institutes here. So they are used to dealing with radiopharmaceuticals, which is very special in this area here in Europe.
Yeah, thanks to the both of you. So we have one question here as well, which I not sure, Falco or Konstantina, would you, if either one of you would like to answer here? We have a question that says, you mentioned metal analysis using ICP-MS and LC-MS. Can you perform mass spectrometry on radioactive material directly?
Should I answer, Falco, or do you want to take it over? Yes, we can perform. We have also projects that are running at the moment, and we perform LCMS analysis in radiopharmaceuticals.
Perfect. Thank you. So yeah, please, whenever questions come in, just let us know so we can answer them after the webinar for sure. So I have a couple of questions here as well. One is, which is more a general question, so maybe Falco, you would like to address this. What are typical timelines for a radio labeling project, which you would place here with us?
Yeah, I mean, it's strongly dependent on the scope of the project. I would say for easy development and validation projects, it's like a couple of weeks. But I mean, if we face a very, very large project from the whole design cycle until like routine testing, it's like maybe 6 to 9 months, but it's really strong dependent on the project scope.
Okay, great. Thank you. And one more question I have here is how early should radiolabeling be considered in a drug development program? So yeah, Konstantina, would you like to answer here?
Yes, how early should the labeling be considered? Yes.
Yeah.
I would say ideally as early as possible. As we want to have to set realistic timelines, and for that we need to log our labeling conditions and the analytical methods that are required as early as possible in order to avoid unexpected challenges or problems that are occurred during method transferring, scale up, and all these things I mentioned. Yeah. So as early as possible is my answer.
Yeah, perfect. And I see one question, Dirk, I think that would be one you could answer. From which clinical phase onward do you recommend validated analytical methods?
I'm not a regulatory GMP person, but if I'm not wrong, it's mandatory from phase 3, and so phase 1 and 2 there is written in one guideline that methods should be scientifically sound. So I would recommend to start as early as possible with a method development to have scientifically sound methods that you only have to validate but not to change. So a very, very, very precise and good method development is the basis for a later validation of the method. You might need it. I'm not sure. Maybe we can look it up and write it in the notes on that. I think it's mandatory from phase 3, but I would strongly recommend to have scientifically sound, proper development method as early as possible, phase 1.
Yeah. Perfect.
You don't want to have to change the method in phase 3.
Yeah, that's true. Well, then, yeah, thank you very much. And as it's 1 minute to 4, so I'm, yeah, want to, show the last slide actually. And thank you again for joining. So it was a great webinar. Thank you to all of our speakers. Thank you for joining. Thank you for posing all those questions. So just be sure we will address them in the follow-up. And so, yeah, we are really happy about this community because it will really, yeah, bring us closer to our joint goal, which is safe radiopharmaceuticals for patients. And I think, yeah, as we always say, we get out of bed in the morning really do that, and we are really happy that all of you took the time to support us here. And and on the last slide we will share that afterwards. You will find our contact data again, so if you have a specific question or something should have been missed, or you want to address another question, just let us know, and please reach out to us directly. So thanks everyone. Have a good afternoon or morning or day. Bye.